Hi everyone, I hope you're having a wonderful day so far. My name is Alyssa, and today my question for you is, how do detectives solve mysteries? Well, it comes from a core science called forensic science, which we'll dig in a little bit deeper today. And today I have a super cool science experiment where we're actually going to extract the DNA sample from a strawberry, something you already have in your house most likely. So, are you curious about crime shows and detective work? Are you curious about how DNA is formed or how we're even going to be able to get this out of a strawberry? Well, hold on to your questions. We're going to do some background review and then we're going to jump into our experiment. Ready? Here we go. You're probably wondering how DNA associates with solving crimes. What is this forensic science Alyssa was talking about? Well, we're going to go over some definitions and then we're going to jump into how all these different pieces correlate to our experiment we're going to be doing in a few minutes. Now, forensic science is the application of scientific principles and techniques to matters of criminal justice, especially as relating to the collection, examination, and analysis of physical evidence. DNA is short for deoxyribonucleic acid, which is the molecule that contains the genetic code for organisms. STR analysis, or short tandem report analysis, is an informative approach to genetic identification and is commonly associated with DNA testing in forensic laboratories, paternity disputes, or missing persons cases. Now, forensic science is a lot like a game, and it kind of involves finding clues, piecing together a puzzle, and solving mysteries. Now, forensic science uses a lot of science to solve legal issues within the civic and criminal justice systems, including the preservation and reconstruction of evidence after time has passed. So, forensic scientists are actually the ones who take, say, at a crime scene, there's a dead body. They're the people who end up taking the samples to help piece the puzzle back together and help solve the mystery. Now, forensic scientists collect and analyze physical and chemical evidence to investigate crime scenes. They use techniques and principles from biology, chemistry, physics, and other scientific areas to help provide evidence which can sometimes be used to testify in courts. They can also specialize in specific areas such as analyzing different crime scenes, running tests in a lab, analyzing DNA, or even bullets. Now, you're probably wondering, how does someone become a forensic scientist? Well, generally, forensic scientists all have an undergraduate degree in a scientific field such as chemistry or biology, or it could be more targeted like a forensic science degree. They can also go on to have graduate degrees, and medical examiners, or MESs, usually have a degree in medicine, which allows them to have a general consensus of how the body breaks down, or different medical terminology that comes into play with some of these crime scenes, as well as some of these forensic scientists as well. Now, DNA is made up of long strings of 4 different compounds or base pairs, also called nucleotides, in a particular order. Now, nucleotides are molecules made up of deoxyribose, a kind of sugar with 5 carbon atoms, a phosphate group made of phosphorus and oxygen, and a nitrogenous base. The nitrogenous bases are adenine, thymine, cytosine, and guanine. Now, all of those different molecules end up being created into a shape called a double helix, and it almost looks like a ladder twisted into a spiral. And the unique thing about DNA is that in each cell of an organism, it actually tells the cells what proteins to make, and DNA is actually inherited by children from their parents. So everyone has their own unique set of DNA, except for identical twins. Now, DNA can be collected from a wide variety of things, from your hair, saliva, tears, sometimes even skin pieces fall off. It's actually really cool how things are broken down and how DNA kind of helps piece the puzzle together as forensic scientists go through and try to solve a crime and help pull all the information together. Now, the STR analysis is can include a repetitive sequence of elements from 3 to 7 base pairs in a length scattered throughout the human genome. By amplifying and analyzing these polymorphic loci and then comparing the resulting STR profile to that of a reference sample, the origin of the biological sample, such as cells or tissues, can be identified or verified. So essentially, the STR takes just a piece of that DNA and looking at repetitive patterns to see if they can find a source of someone, find the correct person from that analysis. And this is taken from their cells, tissues, the DNA that we're going to be pulling. STR looks at 13 of those repeating sections, and the odds of 2 people having the same exact base pairs in all 13 are about 1 in a billion. Now, that's about saying there's only about maybe 6 other people in the entire world who would have the same STR profile as you, which is why forensic scientists like using this STR analysis, because it helps narrow down their crime scene investigations, such as who killed Joe on Friday night based off of the sample they're able to pull from either a murder weapon or figuring how long a body's been dead for. It's really, really cool. Another thing forensic scientists like using this test is because it could take less than an hour and a half to run. And in the crime scene world, time is money. So the faster we can get information to help process and eliminate our candidates of who did what, the better off we are. Other uses for STR include verifying tissue sample origins, authenticating cell lines, deleting cell or tissue mixtures, determining twin zygosity, and tracking genetic mutations in search of studies of diseases such as cancer. So there's a lot of really cool things this STR analysis can help us figure out everyday items, such as whether or not you're going to have say one day you have twins, and then you're trying to figure out if they're fraternal twins or identical twins, or trying to see how genetic mutations affect different cancers, which I think is incredible. So all of this falls under forensic science, which is a very broad and large area. And one of the things that correlates to this is, like we're going to do, potentially solving a crime. This one, we're just trying to pull a DNA sample from a strawberry. But say we needed this to solve a crime, this is one way we would be able to analyze and gather our data. Now, we're going to be extracting DNA from a strawberry. So the materials we are going to need are 2 teaspoons of water, a 1/4 teaspoon of dish soap, a pinch of salt, about a cup of rubbing alcohol. The rubbing alcohol will need to sit in the freezer for about 30 minutes. This is to help the basis of the experiment go well. We're going to need a strawberry, a Ziploc bag, paper towel. You could also use a cheesecloth or a wire mesh. Paper towel might be easiest if you have those on hand. You're also going to need a skewer. This is going to be used to pick up the DNA, but you could also use a toothpick, a fork, or even a sharpened pencil if you have one laying around. On top of those materials, we're also going to need some supplies, such as a glass cup. If you happen to have a science experiment kit and you have a round bottom test tube, go ahead and use that as well. If you're going to use a test tube, you definitely don't need a cup of rubbing alcohol. You're going to need a little less than that. We're going to need a rubber band. I'm probably going to use a hair elastic because I have that on hand. And then we're going to need some measuring spoons as well. All right. So we have our materials and supplies. Let's jump into the experiment to see how we're going to pull DNA from a strawberry. All right, everyone. Now that we have gathered all of our materials into one spot, most likely in the kitchen or wherever you have enough space or access to water, make sure you double check with mom and dad about before grabbing materials and that you're allowed to do this experiment. Even have mom and dad jump in and help you with the experiment. I'm really excited to see the results. So starting off, I have already put my rubbing alcohol in the freezer for 30 minutes. It was actually a little over the 30 minutes. but we just needed it cold in order to help pull the extraction out. So after we do that, we are going to take our Ziploc bag and we are going to add 1 strawberry into our Ziploc bag. Close up the Ziploc bag. And now that it is in the bag, we are going to mash it and make sure it's all squished up and ready to go. So this is probably going to be super fun, squishy. Also, as you're mashing the strawberry up, make sure you're doing the mashing closer towards the bottom of the bag. The closer you do it to the top where the seal is, the more likely it's going to spill out and make a bigger mess. And I'm sure mom and dad don't want to necessarily want to clean up mashed strawberry off the countertops. All right, so there is my mashed strawberry. We're going to hold him off to the side right here. We are now going to take our glass and we're going to start making our extraction solution. Now, in order to do that, we need to get 2 teaspoons of water. So I'm just going to walk over to my sink and fill up my water. Once you have your 2 teaspoons of water, we're going to do our 1/4 teaspoon of dish soap. All right. We're also going to add that into our glass. Now, the dish soap actually helps open up the strawberry cells in releasing the DNA from the strawberry. So that's why we're adding that. And we are now, once we have added our water and our dish soap, we are going to go in and add a pinch of salt. Now, a pinch is just kind of a little bit. Use your best judgment. You don't need a lot. Just a little bit. Whoops, I might have just done a little too much. So I'm going to pour some out. All right. All right. So I've probably added probably a little more than a pinch in my teaspoon cup. I'm going to add that into my mixture. And now once we have that, we're going to mix it all together. So mix, 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 mix. And the thing that the salt ends up doing, it actually helps the strands of DNA clump together and it helps make it visible to the naked eye. So we'll be able to see it without having to look through a microscope, which is awesome. All right, so once we have this all mixed up, ready to go, now that I can't see the salt particles anymore, we are gonna reopen our mashed strawberry bag. We are going to pour our extraction into the cup. All right, and now we are going to seal it back up, and then we are just going to make sure it's well mixed. It did say that this might take a couple minutes, so take your time, make sure it's fully mixed in together. So we want to make sure that the strawberry is able to take the extraction solution and that we're going to be able to visibly see our DNA once we're all ready to go. All right. So we are mixing, we're squishing. And again, as we're mixing and squishing the extraction solution into the strawberry, make sure you're doing it more towards the bottom of the bag. That way you don't make them, just in case the seal pops open up the top, it doesn't make a mess and you have more to clean up afterwards. All right. I say that looks pretty well mixed up to me. All right. So we are going to take our glass we just had. we are going to add our paper towel on top. Take your rubber band, or I'm going to use a hair elastic in this case. Place it over the top. And now that we have our rubber band on top, we are going to gently kind of make a well. So gently push the paper towel in. So it kind of makes like a little bit of a cup up at the top. So almost like a little hole up the top. And once we have that, we're going to take our strawberry and extraction solution. We are going to carefully ask mom and dad for help if need be. Carefully pour it on top of this. And now once we have all of this sitting on top, we're going to squish it out right over here. All right. And it's said that this might take a couple of minutes. So I'm going to let this sit for, it said anywhere for 3 to 5 minutes. So I'm going to let this sit for 5 minutes, and it's going to drip some of the extraction solution into the cup. So once we have our extraction solution into the bottom of the cup, as you can see, we're already starting to get stuff down at the bottom. So let's leave it for a couple minutes, and then we'll come back and finish the experiment. All right, so it's been a couple minutes. I've let this sit probably a little longer than 5 minutes. So I am now going to carefully remove my extraction. Now, I seem to have gotten quite a bit of it into the bottom of my glass. It's okay if you don't have that much in the bottom of your glass. It said you only need like a couple little drops in order for the experiment to work. So now that we have, just cleaning up as I go. Now that we have pulled this off, we are now going to add our cup of rubbing alcohol. I just grabbed a measuring cup. If you feel more comfortable just pouring a little bit in, or double check with mom and dad before you do this. All right, now that I have my cup of rubbing alcohol, I am going to add it into our mixture. Now, we're going to let this sit for a minute, and in the directions it said that we might actually start visibly seeing a DNA strand start floating from the top. Now, I don't have the best glass right now because it has a little bit of ridges on the outside of it. But I'm going to let it sit for a little bit and see if I see any of the DNA start coming up to the top. And as you're doing this, see if you see any of the, if you see the DNA kind of moving and dancing in your cup. All right. Just trying to see if I can see it at the bottom. So, with your skewer, toothpick, fork, whatever you're using to help kind of pull the DNA to the top of the screen, gently kind of go in, see if you can find it, see if you can pull it to the top. Ooh. I actually do kind of see it clumping. It's all kind of towards the bottom of the cup right now. Oh yeah. So mine looks like it's a pretty big piece of DNA, which is actually kind of really super cool. Let's see if I can get this closer and you guys can see it. All right. So as I'm picking this up, you can kind of see on the edge of my skewer, this is the clump of DNA. Now, it doesn't look quite as stringy, and isn't that awesome? Look at how cool that is. So, what is at the end of my skewer, or my chopstick, I should say, is a piece of the strawberry DNA. Or it actually looks like a pretty big piece of strawberry DNA. and you can actually just kind of like twirl it and mix it and kind of just see it float. It's actually really, really cool. So that's kind of a small experiment in how forensic scientists or even detectives are able to kind of pull a DNA sample and be able to solve a mystery. That is incredible. So, as I, try and put this on the cup without it falling off. So, what were your results like? Were you able to see the DNA? Were you able to pull it to the top and kind of play around with it and see how it looked at the end of the stick? Was it as cool as you thought it would be? Is this something you would have never thought of doing, or something you're interested in? Let me know on the website through the comments, and how you thought the experiment went. Was it something cool? Is this something you'd want to continue looking at? Love to hear your feedback. I hope you enjoyed yourself and got to learn another really cool science experiment. I hope you guys have a wonderful rest of your day, and I'll see you guys soon. Bye, guys!